Hello, everyone. Thank you for joining us. My name is Dawn LaPret, and I am one of the program managers at C2ST. Welcome to today's program, Engineering Plants, the 12,000-Year Plan. This program is presented to you by the Chicago Council on Science and Technology in partnership with the Carl R. Rose Institute for Genomic Biology and the Kleinman Center for Genomics in Business and Society. The Carl R. Rose Institute for Genomic Biology is where science meets society. It is an interdisciplinary institute dedicated to the transformation of research and technology in the life sciences. They use team-based strategies to tackle grand societal challenges. If you are new to the Chicago Council on Science and Technology, we are a 13-year-old organization working to enhance the public's understanding and appreciation for science and technology and its impact on society. Please visit c2st.org to learn how you can attend virtual programs in the future and get information to donate. Now for a little bit of housekeeping. Please type c2st2.cnf.io into your web browser and click today's session to ask questions throughout the talk and evaluate the program at the end. You will be able to ask questions throughout the program while you're watching. Our staff will be monitoring C2ST TV, YouTube Live, and Facebook Live. Again, to ask questions, please go to c2st2.cnf.io. Now on to our featured event. I would like to introduce Dr. Lisa Ainsworth. She is the research leader of the Global Change in Photosynthesis Research Unit and an adjunct professor of plant biology at the University of Illinois at Urbana-Champaign. She earned her bachelor's degree in biology at UCLA and her PhD in crop sciences at UIUC. Her research aims to improve the core response to global atmospheric change, including the rising carbon dioxide and ozone pollution on crops. She has held leadership roles in the American Society of Plant Biologists, the International Society for Photosynthesis Research, and the American Association for the Advancement in Sciences. In 2019, Dr. Ainsworth was awarded the National Academy of Sciences Prize in Food and Agricultural Sciences, and she was an elected member in 2020. Dr. Ainsworth, we are thrilled to have you join us today. Welcome.
Very much. I hope you can hear me okay. And I'll start the slideshow in a second. All right, there we go. Okay, so I'm thrilled to be here today to talk to you about engineering plants, the 12,000-year plan. And I think we're going to go back to the beginning with this talk. And so what I'd like to talk about is plant breeding, sort of the history of plant breeding and what it is. So plant breeding is a deliberate effort by humans to sort of nudge nature to our advantage or to our liking. And breeding is about manipulating plant attributes, their structure, their composition, their taste, most importantly, to make them more useful to humans. And breeding works because of the heritability of traits. And so because traits are passed down from generation to generation in plants, just as they are in all living organisms. So plant breeding really started probably 10,000 or so years ago. And so the history of it dates back to 9,000 BC. And so during this period, that's when humans discovered the most basic breeding technique, and that is selection. That's the art of selection or discriminating biological variation in a population to identify and pick the most desirable plants or animals in this case as well. But if we think about deliberately engineering plants, that probably dates back to Thomas Fairchild in 1719. And he was the first to make an artificial hybrid where he took the pollen from a carnation and he then put that onto a sweet William flower. And in doing that, he took the pollen from one organism, put it onto the flower of another organism, and created Fairchild's mule. And you can see those pictures here in this image. So following on from those experiments, Joseph Koelreuter performed the first systematic experiments in plant hybridization. And he did this with Nicotiana species. And so we know Nicotiana as tobacco. So what he did when he did his experiments was he tested the pollen from one species of Nicotiana, and he put that onto the flower of another species of Nicotiana. And he found that the offspring of these crosses resembled both parents. And this was really groundbreaking. So his experiments discovered the importance of pollen in determining the characteristics in the offspring plants. And so really until he did those experiments, the maternal plant was really thought to be the only one that really mattered. And of course, you know, these experiments weren't believed by many of his contemporaries at the time, like lots of science is not. But his experiments did discover the importance of pollen, and they also were some of the first to discover that the progeny of a cross, so the children that are derived, or the small plant, the child plants that are derived from the cross of two parent plants, are often more vigorous than the parent plants. And this will come up later as well. So in the 1960s, Gregor Mendel used mathematics to explain the results of crosses in a quantitative way. And so he was really the first to predict the concept of genes in plants, and that genes occur in pairs, and that one gene of each pair is present in the gametes. And so from the male flowers and the female flowers, and the eggs and the sperm in plants as well. So if we move forward a little bit from Mendel, Wilhelm Johansen in 1903 developed the pure line theory. And so this was really important to plant breeding as we know it today. And so what Johansen confirmed was that if you back cross a line, you can produce a uniform true breeding cultivar. And so if you take the progeny of a cross and then you back cross that to one of the parents, and you do this repeatedly, you can get a true breeding line, and where all of the genetics are fixed in that line. And then that allows geneticists to select for the most important traits, and they're no longer segregating in the progeny. So next, George Shull studied this in maize. And so this was incredibly important, especially to the history of our country and the state of Illinois. And so Shull studied hybrid vigor in maize, and he was the first to coin the term heterosis. And so what he showed is that if you have two inbred maize lines that maybe have yields of about 20 bushels per acre, and you cross those, the hybrid offspring produce about 80 bushels per acre. So this is a four-fold increase in yield in the offspring. And this is the concept of hybrid vigor. In his 1946 science paper where he talked about his work, he showed some of the crosses that he made. And so when he crossed strain A by strain B, he got this F1 or filio 1 generation plant, and it had about 4 times the yield of either parent. And then he showed his initial work that he was doing in the early 1900s, in contrast to a commercial hybrid that was really developed, and the whole hybrid seed corn industry was developed based on the work that was done by Shull. And I think the importance of this work can't really be understated. So from just 1942 to 1945, this was early in the adoption of hybrid corn in the United States. Hybrid corn produced 1.8 billion bushels in those years from 1942 to 1945. That increased the value of corn in the United States to $2 billion. And so when Dr. Stadler from the University of Missouri testified before Congress at that time, he said that it was no exaggeration to say that the United States' investment in the research on hybrid corn was enough to more than pay for the cost of the development of the atomic bomb. So, you know, this was really an incredible example of how investment in research pays off. And of course, we see that now. We've seen sort of the results of this investment in research. And this graph is showing you corn grain yields in the United States. And so before Scholl did his research and really identified the importance of hybrid vigor, yields were around 20 to 30 bushels per acre. And then once we started using hybrid lines, yields went up, and then they've just gone up tremendously as a result of both breeding, so better genetics, as well as better management in corn. And so this is really a tremendous increase in yield. And when yields were going up in the United States in corn, the Green Revolution was also happening around the world. And so the Green Revolution symbolized the modernization of agriculture in the 1940s and the 1960s. And Norman Borlaug was the one who really took this to less industrial countries and created sort of a technological change there. And so the key things that Norman Borlaug did in his work was he bred dwarf varieties of grains, and so that means that they were shorter, and so they were less likely to fall over or lodge. And he also developed lines that could really use nitrogen fertilizers. And so in that way, you had a short-statured crop that could invest a lot of nitrogen in its grain and have very high yields. And he won, of course, the Nobel Prize for this work in 1970. And so this graph is just showing you some of the gains from breeding in wheat. And so you can see wheat yields were about 2 tons per hectare from the late 1880s to about the 1940s, and this is in the United Kingdom. And then with the Green Revolution, you had this tremendous increase in wheat yields. And in the UK today, the yields have staggered a bit. And if we think about breeding for the future, we need to breed crops to withstand higher temperatures, to withstand drought stress, and also to use nutrients a little bit more efficiently, because one of the downsides of the Green Revolution has been there's been a tremendous increase in pollution due to agriculture. And so what does this mean if we're trying to breed for the future? We have to understand a little bit more in detail the process of breeding. And so if we think about wheat a little bit more and think about the wheat genome, we can think of the genome in terms of a book, right? And so if the wheat genome, if we think about it in terms of the book, one page of the book might contain 10,000 base pairs. And so these are the nucleotides that make up the genetic code. And that might contain about 2 genes in those 10,000 base pairs in a page of a book. And so we'd need about 1,700 books with 1,000 pages each to have sort of a record of the wheat genome. And so that would be 1,700 books or 1.7 million pages or about 17 billion base pairs in total. And this is roughly 5 times the size of the human genome. So in breeding, what happens is you have, say, one variety of wheat that's represented by the red books, and maybe that's a really high yielding variety. And there would be a second variety of wheat, and maybe this one has a trait that makes it tolerant to drought stress or high temperatures. And you cross those varieties, and then you get random retention of information from each parent. And so this would be for sort of the start of hybridization or crossbreeding. In a conventional wheat breeding program, you would take a donor parent that maybe has a gene of interest. Maybe it's a temperature-resistant gene or a drought-resistant gene, and you cross it to an elite parent, and you get the progeny, and half of them kind of have attributes of the elite parent, and half of the alleles look like the elite parent, and half of the alleles look like the donor parent. Well, then you would cross those back to the elite parent, and you would cross them back to the elite parent, and you would do this over and over again, and this is the process of back crossing. And the idea here is to hold on to as much of the information that's in the elite parent that you like, so high yield and maybe lodging resistance, etc., and only hold on to the trait that you're trying to cross into there from the donor parent. So in this case, maybe temperature resistance or drought tolerance, something like that. And so for a conventional breeding program, you would start with selected parents, and you might say make 800 crosses. That would give us 2 million plants to look at if we're looking for a trait of disease resistance or drought tolerance or temperature tolerance. And then in the next generation, you would look at 400,000 plants again for the traits that you're interested in. And each of these generations, we're going down 11 or so generations to get to an elite line. And so this takes quite a lot of time. This process takes time. And so you can maybe speed it up a little bit by going to nurseries in different parts of the world, but it is still a slow process in terms of the breeding. That said, that slow process has provided us with the incredible array of foods that we eat today. It's also provided us with resistance to important pests. And so just for example, in potato, the Phytophthora pest is the one that caused the Irish potato famine, and we have resistance to that today. We have powdery mildew resistance in soybean and rust resistance in wheat. Plant breeding has also achieved adaptation to a very wide range of latitudes and growing conditions. And so just for example, soybeans can grow from Canada to Florida, and beans are adapted to each of those different environments with different day lengths. We can also use mutagens or agents that cause damage to DNA and cause changes to the genes to create greater diversity in our foods. And so one example of this is that the ruby red grapefruit variety Rio Red that was released in 1988 was developed by radiation of seedlings. And so by radiating these seedlings, there was a spontaneous mutation in an enzyme that causes that converts the red pigment into sort of a colorless pigment. And so these lines that had the radiation stayed red and created the ruby red grapefruit. So as we look to the future in our pathways to modify crops and engineer them, we can find and use natural genetic diversity in our breeding programs, and that's what we've been doing for really for 10,000 years. As we've been selecting plants, we've been using the natural variation that we see and identifying the lines that are most useful. But now, using genomics, we can also design or move genes into plants that we want to use. And with the help of bacteria, we can introduce genes into plants and then change very specifically the nature of the plants. And we're doing this. And so some of the most important genetic modifications in terms of agriculture that have already been introduced to plants are the engineering of Bacillus thuringiensis into plants. And so BT is a potent toxin to Lepidopteran larvae, and so these are caterpillars of moths and butterflies. And so when the pests eat crops that have the BT protein, they eat less because then they can't digest it. And so this has been engineered into corn, into soybeans, and it's although a little controversial, actually quite controversial because of off-target effects, this genetic engineering has been widely adopted into agriculture today. Another very widely used genetically engineered crop is herbicide resistance. And so by engineering the ability of crops to withstand herbicides, you can spray the field and the herbicides will kill the weeds, but not the crops. And so these two modifications, BT and herbicide tolerance, have been widely introduced. And so almost 100% of acreage in the United States is now in genetically engineered soybean and corn. And this is just showing you some data across the globe. This is the area of biotechnological crops from 1996 to 2017, and this is in million of hectares. The blue line is showing you industrial countries, the yellow line showing you developing countries, and then the green bars are total. So today, well over 180 million hectares across the globe are planted in biotech crops. And so in the United States, most of the soybeans are biotech. And so if we look in the United States specifically, for soybeans, 100% of the soybeans have either herbicide tolerance or this isn't in Argentina, sorry, 100% of the soybeans grown in Argentina have herbicide tolerance or insect resistance. And in the United States, it's over 90% of maize that has herbicide tolerance and or insect resistance breeded into it. So there are other crops that have also sort of been saved by genetic engineering. And so in Hawaii, papaya is an important crop. And starting in the 1950s, the papaya industry began suffering from the impact of the papaya ring spot virus. And that looks like this. This is a papaya that has the ring spot virus. And so at first they just moved on which island papayas were grown in Hawaii. But then it was clear that it was a bigger problem than that. And so in particular, Dennis Gonsalves started working on developing resistance to papaya ring spot by genetic engineering. And this was critical to use genetic engineering in this case because there was no natural resistance to this ring spot virus. It was a potavirus, and there was no natural resistance in the germplasm. And so using a transgenic approach, he developed a rainbow variety of papaya that is resistant to the papaya ring spot virus. And now today, over 75% of the acreage in Hawaii uses this genetically modified line. And so with the advent of genomics, we also know a lot more specific information about the genomes of our crops. And so we have now genomes of many plant species that have been sequenced, and this ranges from model species to crops to trees. And we have new tools in our bag where we can more specifically engineer the genome. And so no longer do we just need to rely on crosses where we're randomly retaining the genetic information from each parent. Now with the tools of genomics, we can more specifically engineer different genes. And so an example of that is in rice. And so rice was genetically engineered using CRISPR technology to produce more favorable oil profiles. And so rather than having high oleic, or the rice was produced to have high oleic and low linoleic oils, making it more healthy. At the University of Illinois, we're using genetic engineering to improve photosynthesis in plants. And so this is work done by my colleagues in the Genomic Ecology for Global Change theme in the Institute for Genomic Biology. And so photosynthesis evolved perhaps not to maximize yield of crops. And so the strategies that my colleagues and I are taking to improve photosynthesis using genetic engineering are manyfold, but the examples that I'm showing you here are specific modifications that allow plants to be less sensitive to changes in light and to respond more quickly when plants go from shade to sunlight to take full advantage of the light. And also to the second pathway, the synthetic glycolate metabolism, is to take a bacterial pathway and put it into the chloroplast of plants to improve the efficiency of photosynthesis. And so these are just two examples of sort of the future of where we're going with genetic engineering in crops. But there are many others, and so this is just showing you some examples. There have been scientists who have been looking at resistance to all sorts of new and developing diseases. As I mentioned before, key targets are improving the water and nitrogen use efficiency of crops, and so crops become more sustainable. And when we think about sort of the nutritional aspect of foods, there are many foods where you could increase their nutrition by, say, expressing a new biochemical in those plants, but you can't find those biochemicals naturally in the genetic diversity of the species. For example, the pigment that causes the purple color in eggplant is very healthy, but it's just not present in tomatoes. But you can engineer it into tomatoes. You can introduce it from eggplants into tomatoes and produce purple tomatoes that have health benefits. And so there are challenges of delivering these genetically modified traits into plants. And one of those is just the time. If we think about the time for agricultural innovation, it's about 28 years. And so, that's one of innovation per career for a scientist. And so that's not fast. And genomics and genetic engineering has the potential to decrease that time. There are other concerns about intellectual property, sort of the freedom to operate, and licensing. Always the economics of whether a trait is going to be valuable or not is important to consider, and a limitation. And there's also consumer and societal acceptance. And so even traits that have potential benefits aren't necessarily accepted. And golden rice is sort of the poster child for that story. And so I guess I'll just leave with, genetic engineering in crops, something old and something new. So humans have been altering plant genomes since the dawn of agriculture. And advances in genomic technologies and understanding have the potential to speed up and expand our opportunities for genetic engineering of crops. So that's the end of my presentation, and I'm happy to take any questions.
Oh, thank you so much, Dr. Ainsworth. We do have a number of questions. It's a nice variety. I would say one of the most popular questions that have come up would be in regard to CRISPR. And the question is, does the CRISPR adapt easily to the plant system, or are there plant-specific challenges when you're using it?
Yeah, so that's a good question, and there are challenges for using it in plants, but there have been a number of success stories, and so people are successfully using CRISPRs, and there are a variety of them that are used today in plants.
Okay, thank you. Another question is in regard to climate change and how that influences the work that you're doing.
Right, and so my specific research it aims at adapting plants to climate change, and so I study how plants respond to rising carbon dioxide concentrations and also ozone pollution. And so I think the elements of climate change are really providing both new challenges to agriculture and new opportunities. And so if we can identify germplasm or engineer plants to take advantage of, say, rising CO2 by increasing their photosynthetic rate and maintaining their nutritional quality, as well as be tolerant to increasing temperatures and drought stress, that's really the challenge that we have. And so oftentimes there's a trade-off between breeding for one thing and then sacrificing something else. But certainly thinking about the future growing environment in the context of selection and breeding is incredibly important.
Oh, that's good to know. There's a question related to that in regards to when you address one thing, another item might arise. And so, regarding the genetic modifications, for instance, for the insect resistance, have there been instances when, let's say, the insects adapt to this crop resistance that you intentionally created, and then are new modifications necessary?
Yeah, and I think probably the clearest demonstration of that is with herbicide tolerance. And so, you can engineer plants so that, say, they break down glyphosate and they're tolerant to one chemical. Glyphosate's a good example. That's the one that's in Roundup, right? That everybody all over the world is using. And we read a lot of news reports that glyphosate's in everything. So we've engineered corn and soybeans and cotton so that they're resistant to glyphosate. But then we've gone and sprayed the world with lots of glyphosate. So now lots of weeds are resistant to it. And that's kind of an arms race. And certainly the same is true for pests and plants. And so, plants and the pests that eat plants are engaged in sort of a biochemical arms race, and have been since plants or pests have eaten plants, or insects have eaten plants. And so I think, yeah, we can look at this in the context of sort of natural evolution, as well as in the context of selection in agriculture.
Okay. All right. Thank you. There are a good deal of questions regarding your experiences as a female in science. And one of the questions more directly was, can you touch on your experience as a female in plant biology? What struggles have you faced and how do you persevere?
Um, that's also a really good question. And so I guess my work straddles a lot of different disciplines. So I'm a plant physiologist by training. I'm in a plant biology department in the School of Integrative Biology at the University of Illinois. But I'm also an agricultural scientist. And so females are represented at very different levels in those different fields. And so in biology, I have very strong and positive female role models, and there have been plenty of them throughout my career. And so I think that, that's an area where I've felt supported. Crop sciences is quite different. Agricultural sciences is still not well represented with women, especially at the sort of level of professor and higher. And so I think, I think that there are historical reasons for this, and I think that I guess in my career, I have seen a lot of change for the positive in terms of engagement and involvement of women in agricultural sciences and in biology. But I think we have a ways to go, and so I just saw a report yesterday. That the AAAS is working on with 3M, and I think that many people don't go into STEM because they're sort of discouraged at a young age that it's not for them, or they're not smart enough, they don't see a path in it. And I certainly felt that way in terms of pursuing, say, farming. As a young child, I grew up in a small town in central Illinois, and I don't think I was ever encouraged to pursue farming as a career, whereas probably 50% of the boys that I went to school with were, right? And so I think that that's cultural. I think it's changing. I think for women in the sciences, I guess in my career, certainly in biology, I have only seen support for women in science and more women joining STEM there. So I've been encouraged.
Oh, that's so good. I'm really glad to hear that because we know that there are a variety of experiences that women can face when they're in the sciences, particularly at institutes of higher ed. So I'm really glad for you to share that. On a related note, if someone was starting out with a career in plant biology or agricultural sciences, what advice might you give them, given that there may be issues with finding a job in the field? Are you aware of what the career outlook is like right now for a plant biologist?
I think, well, I mean, right now, in the time of COVID, certainly the career outlook, it doesn't look great. And it doesn't look great in many, many fields. I think, academia is struggling right now, and there are fewer jobs there. There are government jobs that are still open. I don't know that the Ag Research Service is expanding right now, but there still are openings. And I guess these are trends that are hard to predict. I think if we look at what's happening in the future in the direction of where sort of technology is taking us, there's incredible interest in sustainability. And I think plants have a huge role to play in sustainability in the planet. There's always a need to increase the efficiency of food production, whether it's for sustainability or whether it's for just improving food security. And so I think the trends in agriculture have certainly been that there are fewer people in agriculture in general in the United States. There are very few people that are still sort of farming. At the same time, the technologies that are available to farmers today are incredible, and there are new trends in using data in a way that we haven't before to farm smartly and to farm better. And so I think that there are a lot of opportunities, but it is difficult to predict that landscape at this moment.
And that's fair. I would say it's difficult to predict the landscape in a lot of areas right now. But we still need to grow crops, we still need to eat, and we still have concerns about what happens with our atmosphere. So thank you for that. A different question in a little bit of a different direction is, sometimes when we hear the term genetically modified organism, GMO, there's this negative reaction or outlook. Can you share your thoughts?
Sure. I think that a lot of that is from not understanding the technology, right? And so when we talk about genetically modified organisms, we're talking about introducing the genes from one organism into a different organism. And so, the examples that I gave of inserting the Bt toxin that is in bacteria into plants, so that when caterpillars eat those leaves, they can't digest the leaves. That was deliberately taking an enzyme from bacteria and inserting it into plants. That is the same, in principle, the exact same technology that is used in medicine to create many of our drugs. So, for example, insulin is created using genetically modified organisms in much the same way. And so I think that when we think about food and when we think about crops, there's also a huge cultural aspect there. And so I think that people have a negative or a negative just reaction to genetically modifying foods in a way that perhaps we don't when we think about medicines. But I think if we break things down to what the science is and maybe divorce the science from the cultural aspects of what we're thinking about, then our reaction may be different.
I see. Thank you. Thank you for sharing that. I think the medicine analogy is really helpful. And sometimes we're so inundated with all of these different sources and these news outlets that are telling us things, and we hear GMO in the headline, and we may not actually understand it, but we take away this, oh, maybe that's not good. And that's exactly why C2ST exists, to bring experts like you in touch with the public so that we can try to demystify some of that understanding and to share what's going on. So it's not just a buzzword that has this negative impact. Thank you. Thank you for answering that. Um, what would you say is the future in plant engineering? Like, what's the next thing or the current thing? I know you're working on crop science. Where is that taking you?
Right. So, I mean, I think that there are a lot of things that we can do better through engineering plants. And so, I mentioned improving photosynthesis. And so, I showed some data where wheat yields in the UK have already sort of started to plateau. And so that's a problem because the population on the planet is growing. That's also a problem because we don't actually want to take more of our natural lands and turn them into croplands. And so we need to figure out how to make crops more productive. And so one way is to engineer photosynthesis, and that's what's the goal of the RIPE project at the University of Illinois. I also think there is a lot of opportunity to increase the efficiency of crops, so their nutrient use efficiency and their water use efficiency. So if we go back to the previous question about GMOs and why people might have a negative reaction to them, well, one reason some people might is because GMOs have also been very self-serving for the companies that have developed them. And so the two traits that are in basically all of the corn and soybeans are insect pest resistance and herbicide tolerance. And so if you're a farmer, you have very little choice but to use some of these products. And they have had some negative impact. And so we do have herbicide tolerant weeds now, and we also have fewer pollinators and many Lepidopteran that are affected by Bt that aren't the target for Bt. It's not that, the target was European corn borer originally, but other we have decreased insect populations for other insects as well. And so
Oh,
I think that if we think about the opportunities in the future, we can learn from what we've done, and we can take advantage of the incredible amount that we've learned from the technology and apply it in a way that improves the sustainability of agriculture. And I think that that will do a lot towards also sort of changing the way that that the public thinks about it.
okay. Thank you. Let me see what we have. Another question that I think was shared, you had mentioned earlier about off targets with these effects of pests. I think you've addressed it in a previous question that we had asked regarding the plant, then the pest then becoming immune, so to speak, to some treatment. But can you say a little bit more about what the off target effects are?
Right. So, well, I guess for example, if you introduce a genetic modification and you're hoping to deter a pest from feeding on the roots of crops, so say the European corn borer, which causes damage to corn yields, you're trying to deter that specific pest, but actually you're affecting all of the caterpillars, not just one specific species. That's an off-target effect,
Okay.
and and that can have huge sort of ecological consequences.
Okay, great. Thank you. Another question that we have would be, have there been findings in your research that you found surprising?
Oh, yeah. I mean, I think that there are findings all of the time that are surprising. And so I guess in my own personal research, I think a lot of the work, as I mentioned, I do looking at how climate change impacts crops, and so how rising CO2 impacts crops. And there have been a number of surprises, and one is that we hypothesized that plants would do better under drought conditions and elevated CO2. We didn't find that. I think of another surprise that was by another group at the University of Illinois was that you expect that pests that eat leaves will eat more of them when plants are grown in high CO2. So in elevated CO2, plants have more photosynthesis and they're making more sugar, and they have lower nitrogen sort of per leaf area because of that, because they have more sugars from more photosynthesis. And so you would expect pests to eat more in that case, and they do, but plants also make less of the compounds that deter pests from feeding in elevated CO2. And that was a complete surprise.
Ah,
And so,
okay.
there are surprises all of the time. Those are two examples, but I think one of the things that have in listening to people who have been involved in sort of genetically modifying crops from the beginning, one of the surprises that you often hear about is just how sort of how difficult it is to get the trait that you want into a crop, and how important lots of things that you don't expect to be important are important. And one example is just the background. So the variety that you are engineering a trait into matters a lot. And so I think that's a surprise and a frustration.
Okay. When you are introducing these traits, I know earlier you had said that sometimes there's a 28-year cycle for an entire strain, and it's a career of a scientist. So is it sometimes just one modification at a time, or do you, based on previous findings, tackle something in pairs? How would you go about um, moving forward with your discoveries? Is it, with it being time sensitive, we're bound then by the amount of time that's available and how fast things can happen?
So it happens slowly for a lot of reasons, and so one of the reasons is that the regulatory process is long. One of the reasons is that it takes a long time for acceptance maybe of a trait, and then lots of things that we try just aren't commercially successful, right? And so this was on average from the time that you're starting, on one idea. So, for example, the photosynthesis work that's been done at the University of Illinois, if we start to sort of the start of that work, maybe 5 years ago, that's 2015, we might expect crop varieties that have improved photosynthesis by 2040. and there are lots of reasons, you know, in addition to the ones that I mentioned. A lot of our research that we do, the fundamental research, is in model species. And so in something like Arabidopsis, which is a weed that has a really small genome and a fast life cycle. And so a lot of the discovery work is in that species. And then in the case of the photosynthesis work that's being done in tobacco, again, that's a species that's quite amenable to transformation. And so you can make changes in the genome. But that's very different from having success in, say, corn or soybean or cowpea or cassava or whatever crop it is that you're trying to engineer. And so that process of going from sort of the initial idea and discovery, testing it in a model species, and then introducing it into a crop. And then even within a crop, you know, there's only so many lines maybe right now that are easy to transform. And so then you have to do all of the work of back crossing it into an elite line as well. And so that's why you know, it takes as long as it does.
That is an incredible delayed gratification, starting with something in 2015 and then actually seeing the yield of that in 2040. Thank you for that perspective. I know that we have an appreciation of things slowly changing over time, but that's certainly some slow change, some slow processes there. Do you have time for one more question?
Sure.
Okay. This question asks about, in regard to plant biology or crop biology, do you feel that this particular aspect of biology is treated differently from the other biological disciplines in the United States?
I think that there's, I guess, I mean, absolutely. There's less funding in plant sciences than there would be in, say, medical sciences, and I think that you know, that's in large part because we have a vested interest in ourselves, right, and in humans. And so, you know, I think there's concern that maybe society as a whole is a little bit blind to plants. But at the same time, plants are an incredible system to study. They have a lot of advantages that animal systems don't have. And they also, you know, they have, I guess, in, you know, my love is photosynthesis, and plants photosynthesize. And this is the process that has created the atmosphere that we survive in. It also, you know, plants produce, they are the primary producers on the planet. And so everything that we eat ultimately comes from something produced by a plant. And so I think that there are strong reasons to invest more. And sort of the story about hybrid corn is one. I don't know that in the 1920s and 1930s, when investments were being made in hybrid corn, that anybody could have predicted that the future of the United States, corn and soybeans, would be our number one exports, right, and a huge part of the GDP of our country. And so I think that's just a good example of how investment in basic research, whether it's plant sciences or astronomy, is important.
No, thank you for sharing that. I think you bringing up medicine and our concern for ourselves, very important. And often in our country, we seem to overlook that connection to nutrition and what we're doing with our crops and how that's also going to affect us personally moving forward. Thank you so very much for your time today. And I'd like to thank our audience for tuning in to today's program. This is the point when we need your help. We would very much like for you to complete the survey for today's program so that we can improve as we go forward. So please go to c2st2.cnf.io to complete the survey. That will help us shape the future of programming to come. And as a reminder, if you would like to rewatch or if you actually missed any part of today's program, this recording will be available on C2ST TV, YouTube, and the C2ST Facebook channel. Thank you, Dr. Ainsworth, for a very informative and excellent program. And we'd like to thank our partners, Carl R. Rose Institute for Genetic Biology and the University of Illinois School System. If you enjoyed today's program, please consider donating to c2st.org. You will also find archives of all of our previous programs and some timely STEM-related blogs. Again, thank you for your time today, and stay safe. Ooh. Are you still with us, Lisa?
Yes.